welcome. This is Katie Wester-Neal. I'm here with Holly Dubs, who's going to be presenting about MECP2 and genetics today. So just a brief introduction. She is a senior genetics counselor in the Epilepsy Neurogenetics Initiative and Division of Neurology at the Children's Hospital of Pennsylvania. She has over 10 years of pediatric genetic counseling experience. She provides clinical genetic counseling for patients with a variety of conditions, which include epilepsy, brain malformations, movement disorders, and other neurogenetic disorders, with a particular focus on Rett syndrome and related disorders, such as MECP2 duplication syndrome, CDKL5 deficiency disorder, and FOXG1 syndrome. She's also involved with clinical research for many of these diagnoses, and she's interested in novel gene discovery for unknown neurogenetic disorders and working to obtain a genetic diagnosis for children with suspected neurogenetic conditions whenever possible. That way she can provide appropriate genetic counseling for affected individuals and their families. We are so thrilled to have her with us today. Just a little note on how this is gonna go. If you have questions, please look at on your screen and type them in the Q&A, and we'll hold those and she'll answer questions at the end of the presentation. So please feel free to put your questions there in the chat and we'll be collecting them. All right, without further ado, I'm going to turn it over. Holly, welcome and thank you.
Great. Well, thank you so much for inviting me and asking me to speak today. I'm really excited to give this talk to you guys. I hope that you find the information helpful and useful. I'm absolutely happy to answer any questions that people may have. So let's, I guess, get started. Does that look good to everyone?
That looks great.
All right, perfect. So today I am going to be talking to you about the genetics of MECP2 duplication syndrome. I like to kind of make sure everyone's on the same page. So I wanted to just start by giving a quick introduction of some basic genetics concepts, followed by a brief summary of some of the different types of genetic testing that might identify MECP2 duplications. And then to discuss a little bit about the relationship between the genetics and the clinical symptoms of MECP2 duplication syndrome, including what we know and maybe what we don't know. And then lastly, to wrap up by discussing some of the different reproductive testing options that might be available for families who are affected by an MECP2 duplication. So just to start off and make sure that we're all on the same page about genetic terminology, if you remember back to high school biology, you'll remember talking about cells being the smallest building block in the body. And then within our cells, we have the nucleus, which is basically the control center of their cells. And our nucleus is where all of our DNA is stored. Our DNA is composed of thousands of different genes and is packaged into small units called chromosomes. And so we all have 23 pairs of chromosomes. This is what they would look like if you lined them up under the microscope. The first 22 are the same in males and females. And then the area that we'll focus on, of course, today is on the X chromosome. So the last pair of chromosomes are the sex chromosomes. Females have 2 X chromosomes, males have an X and a Y. And if you zoom in and look more closely at the X chromosome, at the very bottom is where you'll find the MECP2 gene. And when we talk about locations on chromosomes, we have this little kink in the middle that's called the centromere. The top arm is the P or petite arm, and the bottom arm is the Q arm. And these little bands are numbered in increasing order coming out from the centromere. So at the very bottom of the X chromosome is band XQ28, and this is where the MECP2 gene is located. As I mentioned earlier, and as you probably all know, females have 2 X chromosomes, males have an X and a Y, and this becomes really important when we talk about genes that are located on the X chromosome, because in kind of the typical state, females have 2 copies of the MECP2 gene, whereas males only have 1. And this explains why there are some clinical differences when we see changes in the MECP2 gene, be it either a mutation in the gene associated with Rett syndrome or a duplication of the gene, which we'll focus on today. So one of the big differences that influence the expression of genes on the X chromosome is a phenomenon called X inactivation. Kind of the best biologic example of this is thinking about a calico cat. So a calico cat has different fur colors, and calico cats are inevitably female. And so the difference in coat pattern is due to expression of different copies of the X chromosome in different cells in the cat's skin. And so in females, because they have two X chromosomes, this X inactivation process helps to compensate between that dosage difference between males and females. So in any one cell in a female, only one of her two copies of the X chromosome is being expressed at any given time. And it's essentially a random pattern in terms of which X chromosome is expressed and which X chromosome is turned off or inactivated. So usually we see, again, kind of a random 50-50 pattern, but sometimes we can see something called skewed X inactivation, in which one copy is either favorably expressed or predominantly inactivated. And again, this kind of helps to account for some of the clinical variability we can see in females, and also helps explain why many female carriers of MECP2 duplications are unaffected, which we'll talk about a little bit more later on in the presentation. So I wanted to just quickly show a little bit of a timeline about MECP2. And as you know, the discussion of MECP2 duplication syndrome is inevitably tied to some of the discussion about Rett syndrome. And part of that is because Rett syndrome was clinically described back in the 1960s. And so we really have 50 years plus of data about Rett syndrome, whereas the description and our understanding of MECP2 duplication syndrome didn't occur until much more recently. So even though Rett syndrome was clinically described in the 1960s, it wasn't until the 1990s when the MECP2 gene was actually discovered by the team from Dr. Adrian Bird's lab. And then a few years later, in 1999, MECP2 was actually identified as the genetic cause of Rett syndrome. And this was work done by Dr. Huda Zogby's lab. Several years later, in 2004, it was first noted that overexpression of MECP2 in mice resulted in a progressive neurologic symptoms. And then finally, in 2005, we had the first report of an MECP2 duplication in a human. And then in 2010, by Dr. Peters' group, we had the first really comprehensive description of the MECP2 duplication syndrome that was published. So, compared to 50 years of data about Rett syndrome, we're only talking about, 2 decades of data or less talking about the MECP2 duplication. So, there has been great progress made in that time, but there's still a lot that we don't know. But we're fortunate that we have so much data that has come from Rett syndrome research that we can draw on in terms of our understanding of the MECP2 gene when we start to think about, ways to intervene, targeted therapies, targeted medications, things like that. I'm not going to talk about that in too much detail today because that is obviously a whole other topic that we could spend a whole several hours discussing. But, just to kind of put this in context in terms of how much knowledge we've accumulated in actually a pretty short period of time. And just one quick slide on MECP2, kind of what the gene does and what its role is. So MECP2 gene encodes the protein methyl CpG binding protein 2, or MECP2. And the gene is what we call a transcription regulator. So basically transcription is the process by which other genes are expressed. And so essentially MECP2 helps to control the expression of other genes in the body. Many of the genes that MECP2 helps regulate are important for neuron and brain formation. And essentially, MECP2 helps to control the expression of the right gene at the right place and the right time. You can imagine if MECP2 is either not working properly or missing, as in the case of Rett syndrome, or if there is too much MECP2, it can lead to other genes, many other genes, not being expressed at the way they should, and lead to many different downstream effects. And the more we understand what some of those downstream effects are, we can also think about ways we can potentially intervene. So again, I'm not going to talk too much more about that, but wanted to at least give one slide talking a little bit about what MECP2 does. And I want to transition a little bit now to thinking about testing for MECP2 duplication syndrome. So historically, the diagnosis of genetic disorders is also something that, really we weren't able to do with such precise accuracy and with such detail until the mid or the early 2000s. And so until the early 2000s, karyotypes remained the gold standard of genetic diagnosis. And essentially this entailed looking at a person's chromosomes under the microscope. And unless a person has a very, very big difference in their chromosomes, you're not able to see that. So most cases of MECP2 duplication are too small to be identified via standard karyotype analysis. However, in 2004, microarray technology was first introduced, which allows for the detection of smaller and smaller chromosome abnormalities. So the first type of technology was called comparative genomic hybridization, or CGH. That really became available in 2004. And this image here showing these different colored dots basically is what the data from a CGH microarray shows. And it shows if everything is sort of present in equal quantities, you're supposed to see kind of this yellow color of each individual spot of DNA. If there's too much of something, you see the green dosage, and if there's too little of something, you see the red dosage. And so this is a good way to get a better sense of if there were any smaller pieces missing or extra than what could have been detected by a standard karyotype technology. And if you remember back to my MECP2 timeline, this microarray technology launched in 2004, and in 2005, shortly thereafter, was when the first case of an MECP2 duplication was identified. So really kind of corresponded with the ability to do this much more sensitive genetic testing. A few years later, in 2008, SNP microarray technology was launched, which is basically just an even more sensitive version of the test, allowing for smaller pieces missing or extra to be identified and kind of a better, more precise determination of the breakpoints or the exact pieces missing or extra were developed. And that's really kind of still the technology that we use most often today. And this microarray testing is really recommended as the first peer test for the evaluation of children who present with evaluation for developmental delay or autism spectrum disorder. And so really the majority of individuals with MECP2 duplication syndrome will be identified by chromosome microarray. And again, kind of the benefits of this test are we can really hone in on the exact breakpoints, know the exact size of the duplication, know exactly which genes are involved in addition to MECP2, and get a very close look. And there are some other tests that can identify MECP2 changes. So testing for single gene disorders really came about in 1977 was when this technology really became available. But this technology really is designed for finding changes within genes. So kind of sequence changes or spelling changes within the gene. So this is a good test for finding the types of genetic changes in MECP2 that cause Rett syndrome, but typically other types of technology are required to find small deletions or duplications. And when we think about this type of single gene testing, you really do need to have a high level of clinical suspicion for what gene to test for. And it may be very difficult to recognize mild or atypical cases. And so although MECP2 testing is recommended by the American College of Medical Genetics and Genomics as part of the diagnostic workup of females who present with autistic symptoms, there's not really any specific recommendations sort of targeting the MECP2 gene for males. And so most males, unless you have a very astute clinician, are probably not going to be ordering targeted MECP2 testing. However, next generation sequencing is a genetic testing technology that came about in the 2010-ish. And this testing really allows for thousands or millions of DNA sequences to be run simultaneously. And so with this type of technology, instead of having to do single gene testing, this allows for faster and less expensive testing, and also allows for multiple genes to be tested at the same time. And so with this technology, bioinformatic tools also allow for the detection of copy number variants or deletions or duplications of genes. And so this could potentially allow for testing or identification of MECP2 duplications. This really led to the development of what we call panel testing that allows for simultaneous testing of multiple genes associated with common or overlapping clinical symptoms. And most of these panel tests now often will include deletion or duplication analysis. So if we think specifically about the MECP2 gene, the MECP2 gene shows up on gene panels that are marketed as Rett syndrome or Angelman syndrome panels, epilepsy gene panels, autism gene panels, intellectual disability panels, microcephaly panels, ataxia panels. And so, if a child's clinician orders any of these types of panels for their child for some of these symptoms, all of which can be seen in MECP2 duplication syndrome, this could be another way that children are diagnosed. The kind of limitation of panel testing is that for the most part, depending on the exact technology and the exact lab, the panel will often tell you that MECP2 is duplicated, but it usually won't tell you how big the duplication is, what other duplications, what other genes might be included. And so often, if your child didn't already have a microarray and they were picked up via panel testing, often they might require a microarray as a follow-up test just to really kind of better define the duplication. And then finally, I wanted to talk quickly about exome sequencing because many providers are kind of focusing on this, and this is kind of what we consider to be our most comprehensive genetic test, but it does still have some limitations. This is a test that sequences the 1% of our genetic makeup that really leads to protein expression. So it encodes the portion of our genes that encode proteins, and it's where we think about 85% of genetic changes that cause diseases are found. So this is a test that instead of a doctor needing to think about what one specific gene they want to test or what one panel they want to pick, this allows for testing of all of a person's genes to be sequenced at one time, and it's usually done as a trio where the child is tested simultaneously with a DNA sample from both of their parents. The benefit of this type of testing is that providers don't really need to know what they're looking for. And so it's a good test for children or individuals in whom a genetic disorder is suspected, but either they've already had extensive testing done with no genetic etiology found, or their features aren't really consistent with a recognizable syndrome. And so choosing a targeted test is very difficult. Depending on the lab and the technology, exome sequencing testing may or may not include copy number analysis. Labs are getting better at including this. And so I think there are some guidelines which are recommending kind of moving towards exome sequencing as a first tier test. And so, when we're thinking about the MECP2 duplication syndrome, it's just really important to think about whether or not the exome sequencing will identify copy number variants or not, because if not, then the MECP2 duplication might be missed. If a child hasn't had a microarray already, their doctor jumps right to exome sequencing. So just kind of something important to keep in mind. So I want to just talk briefly about genetic testing results. A lot of reports will include a lot of kind of numbers and letters and what does this all mean. So I've included kind of two different clips of some terminology that you're likely to see from a microarray report, again, since that's the most common test that we'll find MECP2 duplication diagnosed from. And if you look on the left, we'll see the ARR basically just means this is an array result. This GRCH37 is basically the browser version that was used to kind of generate the genetic testing report. So there are different, almost like what version of a smartphone a person has. It's basically what version of genetic data was used to write the report. So that way, when you're kind of looking back at those results or looking at kind of genetics websites, the doctor or genetic counselor kind of knows which version they should be using and which version they should be referencing so that they're pulling accurate material. The XQ28 denotes the chromosomal position that's involved. So in some kids who have a larger deletion or duplication, you might see XQ28, XQ27, indicating that the duplication spans between both of those chromosomal bands. But basically, this really refers to the chromosomal position. And then these big numbers here refer to the exact breakpoint coordinates of a child's duplication. So basically, if you zoom in to the DNA level, this tells you exactly which spot at the DNA level is duplicated. And again, genetic counselors or physicians can then use that data to determine exactly what genes are involved, things like that. And then here, the ×2 indicates the dosage number. So in a male, you should only have 1 copy of this region. So 1 would be normal. 2 means there's an extra copy or it's duplicated. Rarely you might see that say ×3, since there are some children who have triplications or 2 extra copies of this region. And then in some cases, if follow-up parental testing was performed and we know that a variant is inherited, you'll see that indicated by an MAT following this, meaning it's maternally inherited. And then over on the right, again, kind of this combination, 224 kilobase interstitial duplication of XQ28. The 224 kilobase includes or indicates the exact duplication size. We measure genomic duplications or deletions in what we call kilobases, or if they lean into the 1000 kilobase realm, then we start talking about megabases. So 200 kilobase interstitial duplication of XQ28. Interstitial refers to that the duplication does not include or does not extend to the end of the chromosome. In that case, it would be called a terminal duplication. And then again, referencing the chromosomal position of XQ28. So this is just kind of what some of the terminology on a child's genetic testing report might be referring to. And if you have any questions about your child-specific genetics report, your local genetic counselor is a great person to ask, and they can put any of those findings in context for you. So, I wanted to talk a little bit now about genotype-phenotype correlations. And what genotype-phenotype correlations mean is basically, can my son's duplication or my child's genetic finding predict their symptoms or severity? So is there any relation between the exact genetic finding that's present and what we can expect clinically? So for Rett syndrome, we know that 8 common variants account for about 50% of cases within the Rett syndrome population. So 50% of children have one of the same 8 variants in the gene. And so this allows us to get some good patterns recognized and allows for some good genotype-phenotype correlation. MECP2 duplications are a bit different. So there are no what we call recurrent duplications that have been described. So it's not like in every child with MECP2 duplication syndrome, we see the exact breakpoints or the exact same size duplication. It's really different in every individual, which makes it a bit more difficult to predict. However, there are some patterns that have emerged that I'll talk a little bit more about. But what we usually talk about when we're counseling families is that this type of information is useful when you're talking about groups of patients, but it can be difficult to ascribe the data to any one individual and to make specific predictions in any one individual case. However, overall, I'll just talk about some of the patterns that we've seen described and some of the things that we found that can influence the clinical presentation. So first is that size does matter. So larger duplications have been correlated with a higher clinical severity score. And so this image here is basically showing different size duplications that were identified in different individuals, ranging from the least severe or the lowest clinical severity score at the bottom to the highest at the top. And you can see the smaller duplications located down here and the larger duplications located up here. However, it's not like there's like a size cutoff that means like, oh, over this point or over this size, you're going to have much more severe symptoms. It's really all a spectrum, and you can see there are certainly some outliers where this deletion is, or duplication rather, is quite large, but really falls towards the middle of the road on severity. And one of the limitations of this study in particular was that they were using the clinical severity score that was designed for measuring clinical severity in Rett syndrome. So it does have some phenotypic overlap, but there are also some measures that are kind of less relevant to MECP2 duplication syndrome. The other thing that really kind of we have to think about is that in most children, their duplication doesn't only include MECP2. There are other genes involved as well. And depending on what other genes involved, this in some cases can influence the phenotype somewhat. So there is another gene that's really kind of right next to MECP2. It's a little bit hard to see because it's kind of small, but here's MECP2 here, and here is this IRAC1 gene right next to it. So in almost everyone with MECP2 duplication syndrome, they also have a duplication of this IRAC1 gene. And it's thought that this gene might play a role in immune dysfunction. And so it's thought that having this gene duplicated as well might have some impact on the immune issues that we see in children with MECP2 duplication syndrome. Are there children who have this gene duplicated who don't have significant immune issues? Absolutely. And so it's not, a one-to-one correlation, but it's thought that that might regulate some of the immune issues that are seen, but it's something that really needs kind of more research done to be able to determine any specifics about that. Some older data has suggested that the inclusion of two other genes called filamin A and lamin A. So you can see here's filamin A here. Oh, and this is actually, I believe, supposed to say L1-CAM. That is a typo on my part. Here is the L1-CAM gene. There is some data that suggested that those genes might be implicated in some of the GI symptoms that we can see, such as the pseudo-obstruction, but other studies really didn't confirm that association. So they saw pretty significant GI issues, even in kids whose duplications did not include those genes. So again, kind of still something that's being explored, but there are some studies out there that suggest that those genes could factor in terms of the GI symptoms. The most recent study by Dr. Peters' group from 2019 focused in on this RAB39B gene, and you can see this is all the way over here. So in children with smaller duplications, they're not going to have this gene duplicated, but some of the larger duplications will. But independent of size, they found in their study that when they compared children whose duplications included RAB39B with children whose duplications did not include RAB39B, those that included RAB39B had more significant microcephaly and worse motor impairments. And we know that RAB39B independently is also a gene that can cause X-linked intellectual disability with some other neurologic symptoms. And so the presence of that gene included in the duplication leads to some more severe presentation in terms of the motor and the microcephaly. And then the last gene that has been kind of looked at is a gene called GDI1, which again is kind of right here. So fairly close to MECP2, but again, you can see that not everyone's duplication will include that gene, and that gene also has independently been linked to impaired cognition and autism. So could that gene also influence some of the neurologic symptoms that are seen? So again, all of this is kind of interesting data that's still being explored, but we're not able to say, if you show us your genetic testing report, whether or not these genes are included or not, we're not going to be able to say like, oh yes, filamin A is not included, so you're not going to have GI issues. We're not really down to that level of specificity. These are just kind of some trends that have been observed in some studies, but again, in some cases, there have been other studies that have disputed some of those findings. Other findings that we've seen that can influence presentation are position and dosage effects. So there was a recent study that came out last year that looked at where the duplication of MECP2 occurred, and they found that duplications that were not in tandem or not where the duplication was essentially kind of back to back. So here having MECP2 and MECP2 in line with one another, that would be what we would call a tandem duplication. Those cases tended to be less severe than those who had a duplication located somewhere else in the genome. So the example on the right, where you can see one copy of the MECP2 region or XQ28 at the bottom of the X chromosome with another translocated or located at the top of X chromosome, that would be considered a duplication that's not in tandem. And those tended to actually be more severe, with the thought being that they may also involve other genes or other genomic regions, might interrupt another gene at the spot where that duplication is kind of inserted. And so those cases tended to be more severe. We also know that unbalanced translocations, where again, this portion of XQ28 is basically swapped with some other chromosome, we know that in females, this can result in the presentation of symptoms, often because these types of rearrangements can lead to differences in the X inactivation pattern. And there are, again, as we know, some rare cases that have been reported where children have not only one extra copy of MECP2 of the MECP2 gene, but a third extra copy. And those triplication cases do tend to have a more severe presentation. So it seems like the position where the duplication is located, as well as the exact dosage, does seem to have some impact on the overall clinical presentation. And then the other thing that we have to keep in mind is genetic modifiers. So, in addition to the MECP2 duplication, a child has the rest of their genetic background that they've inherited from their parents that just leads to normal variation as it would between any other two individuals. And so the differences in each individual's overall genetic background might influence their clinical presentation as well. There might be variants in other genes that interact with MECP2 that might help the protein work better, and then therefore maybe a more severe presentation in the case of a duplication, or might make the gene slightly less effective, which might help moderate duplication symptomatology at some point. And so the evidence that we have to support this is that, we know that there are some families where children or relatives with the exact same duplication might present with some different symptoms or different clinical severity. So, this genetic background or these genetic modifiers likely plays a role as well, although again, we're not smart enough yet to really deduce the exact details of that. We just know it's something that occurs. We can also see in rare cases genetic mosaicism play a role. And this would be an instance where a duplication was present in some, but not all of a person's cells. And in many cases, this can be an explanation for a milder presentation. So you can see kind of from the graphic shown here that in a person with genetic mosaicism, a mutation occurs or the duplication would occur at some point of cell division while the embryo is still developing. So while the child is still in utero, all of their cells are divided, their cells start off normal, but at some point a duplication on the X chromosome occurs, and then any cells that then come from that cell will have the duplication, but the rest of their cells do not. And so depending on where those cells ultimately end up, can influence presentation. So if those cells are all in the brain, again, then you might see just a pretty typical MECP2 duplication presentation. But if not all of their cells are affected, it can be a more mild presentation because some of their cells just have, one copy of MECP2. So thinking a little bit about inheritance, one of the common questions we get, is how does MECP2 duplication syndrome occur? And as many of you likely know or likely understand, the majority of cases of MECP2 duplication syndrome that we see are inherited. And we see this inherited where a child has the MECP2 duplication, and we do family testing and we find that the variant is inherited from that child's mother. There are some cases where these duplications occur brand new, or what we call de novo, but more often than not, they are inherited. And in some cases, the duplication may have occurred new in that child's mom, but in other cases, it may have been inherited from their own mother or the grandmother. And in some cases, we see other more distant family members who may have also been affected. Oftentimes, that might be like a great uncle who had some neurologic issues and who passed away, but who never had any formal testing. So no diagnosis was ever made. But we, when we hear a little bit more about them, we might suspect that they had the duplication. But again, it's not uncommon for us to see the duplications being inherited from an asymptomatic mother. We call women carriers. And if we were to look at their X inactivation pattern, we often see that that's skewed, meaning that we think that the copy of the X chromosome that has the duplication is preferentially inactivated or turned off in most of their cells, allowing for the normal copy of the X chromosome that doesn't have the duplication to be expressed, which is why we don't often see symptoms in carrier mothers. Again, there are exceptions to that, but kind of in general, this is what we tend to think. And I think it's really important to kind of really explicitly state that, even though a lot of these duplications are inherited, this is no one's fault. In most cases, moms have no idea they're a carrier. There may not be any family history. There was no reason to do any prenatal testing or preconception testing to look and see. And so this is no one's fault, nothing that anyone has any control over, nothing anyone does or doesn't do to make happen. And I think, it can't be stated enough. because I know this is something that a lot of families really struggle with, and kind of feeling like they pass this on to their children, but really they had no control over this. So MECP2 duplication syndrome is what we call an X-linked recessive disorder. And this means that any woman who's a carrier has a 1 in 4 or 25% chance of having another affected child. If we think just about male children, 1 in 2 or 50% of males will be affected, and 1 in 2 or 50% of daughters will be carriers. And here's a schematic here that kind of really just outlines all the different outcomes. So a woman has 2 X chromosomes, the man has an X and a Y, and each are going to pass on one of those 2 to their children. So if mom passes on the copy of the X chromosome that has the duplication, and dad passes on his Y chromosome, that would result in an affected son. If mom passes on the copy with the duplication and dad passes on his X chromosome, that's going to result in a carrier daughter. And if mom passes on the copy of her X chromosome that doesn't have the duplication, and dad passes on his Y, that would lead to an unaffected son. And if dad passes on his X and mom passes on her unaffected X, then we have an unaffected daughter. A question that we're often asked is, should my daughters be tested? Really, the recommendations from several professional genetics groups really advocate to defer genetic testing in asymptomatic minors. So obviously, many people really want to know, they want to know if this is something their daughters need to be thinking about when they grow up. But since in the majority of cases, it's not going to impact their medical management, we typically really do advocate deferring testing until they're able to make their own informed decisions with appropriate genetic counseling. There, I think, are a lot of psychosocial implications, a lot of potential insurance discrimination considerations, and things like that that people often just don't think about that could potentially factor in. And so the general consensus is we like to defer testing. There may certainly be some exceptions to this rule that may warrant testing in childhood or an older child with their own informed decision making, but kind of the rule of thumb in general is we tend to defer genetic testing in asymptomatic minors whenever possible. And then lastly, I just wanted to wrap up my presentation today with kind of talking a little bit about different reproductive considerations. This is a topic where people have very diverse perspectives on, and some people, feel that even though there is a recurrence risk, that's not anything that they would feel the need to do any sort of testing or intervene in any way, and that is totally appropriate. But for anyone who kind of is interested in kind of hearing more about what types of different reproductive options might be out there, I just wanted to highlight some of those briefly here today. And so one option that would be available to women who know they're carriers is something called preconception genetic testing or pre-implantation genetic testing, PGT. This is testing that is combined with in vitro fertilization. And so basically a woman undergoes hormone stimulation to get their body to produce more eggs. Those eggs are then taken out of the body, and in vitro fertilization is performed. And so those eggs are fertilized with sperm outside the body. And then in the early embryo stage, the cells are allowed to divide a few times. So at around the 6 to 8 cell embryo stage, one cell is taken and genetic testing is performed to see if the duplication is there or not. And then a couple could choose to use an unaffected embryo to implant to try and achieve a pregnancy. So this is obviously an involved procedure. It's not for everyone. Insurance coverage and costs vary from place to place and from plan to plan, but it's just something that is out there if couples wanted to explore that as an option, as a way to have another child that does not have MECP2 duplication syndrome. Our kind of standard prenatal genetic testing is via invasive diagnostic procedures, and those include chorionic villus sampling and amniocentesis. Chorionic villus sampling is able to be done a little bit earlier in a pregnancy at about 10 to 13 weeks. And in CVS, basically a sample from the placenta is taken, which has the same genetic findings as the fetus, and then sent for analysis to look for the presence of MECP2 duplication or other genetic differences. And then amniocentesis can be done later, a little bit later in a pregnancy at about 16 to 20 weeks, where they take a sample from the amniotic fluid that surrounds the fetus to look for any genetic differences to determine if there are any findings there. And again, that's done a little bit later on in the pregnancy. Both provide accurate diagnostic information. There is a small risk of a procedural related miscarriage associated with both procedures, which is, a risk. But overall, those risks are relatively low, being somewhere around 1 in 100 to 1 in 200 for CVS, around 1 in 200 to 1 in 400 for amniocentesis, kind of depending on your provider and your care center. Testing that's really kind of been gaining traction over the last few years and really kind of expanding in its clinical applications is something called non-invasive prenatal testing, or NIPT. And basically what this testing is that as the fetus is developing, it basically sheds little particles of DNA that can be found in mom's blood. And so basically this is testing where a blood sample from mom is taken and able to do genetic analysis for the fetus. And so really it's become pretty common practice for certain chromosomal disorders like trisomy 18, 13, and 21, and certain chromosomal microdeletion syndromes like 22q11.2. But as labs are getting more comfortable with this technology and with testing, they're starting to really kind of expand in their applications. And so currently, they're able to do some actual genome-wide chromosomal testing, but at this point, the threshold or the resolution is such that they may be able to detect really large duplications that include MECP2. So this could be useful for families where they might have a balanced translocation, where they have the XQ28 region sort of stuck on another chromosome somewhere, and there's really big pieces of chromosome involved. But for most of the smaller MECP2 duplications, this type of technology really just isn't sensitive enough to identify those types of duplications. But that's not to say that in the future we might not be there. We might not be able to do this type of testing for MECP2 duplications. I did also want to point out that testing recently also became available for certain single gene disorders offered by certain labs, and this does include Rett syndrome and the MECP2 gene. But really, this is looking for MECP2 gene mutations that would cause Rett syndrome. It's not going to be testing for MECP2 duplications, and so this would not be an appropriate test to look for MECP2 duplications. Some OBs might be not as familiar with the nuances of that type of testing. So, if they bring that up as a possibility, really, we're not there yet. And then lastly, just because these are things that sometimes people don't always think about, and they're, again, not for everyone, but some families might consider things like use of a donor egg adoption or even embryo adoption, where couples who have previously gone through the IVF process have embryos that they have not elected to use for a pregnancy and offer those to other couples who might be in need. And so these are just other things that are out there and things that people can consider. So in summary, improvements in our genetic testing technology have really led to an increasing ability to diagnose MECP2 duplication syndrome over the past 2 decades. And although there is some genotype-phenotype correlation that exists, this is really complicated by a lot of other factors. However, some of the things that we know do matter include duplication size, position, and genetic modifiers. The majority of MECP2 duplications are maternally inherited, and there are different reproductive options out there for MECP2 duplication syndrome carrier mothers, if that is something that they and their partner are interested in pursuing. So thank you all for listening in today, and I am happy to take any questions.
Fantastic. Thank you so much. Yeah, let's see if you want to take your screen off screen share.
I think I just did.
Did you? Okay. Oh, there we go. Yes.
Okay.
All right. I can see it now. Excellent. So we have one question so far in the Q&A, and I just want to say again to our attendees, if you'll put your questions in the Q&A, we have some that people send in early, and then we are going to take questions live as well. So here's the question. It says, I'm curious how identical twins can have different experiences with MECP2. I understand that they're different people, but my identical twin sons are 90% identical. Would that 10% difference make that much of a difference?
So I'm not totally sure what you mean about them being 90% identical versus identical. I think we kind of identical twins we assume have the same genetic makeup and the same genetic sequencing. One of the things I didn't talk about today is something called epigenetics, which basically are kind of little signals that are put on different genes that kind of impact their expression. We don't do routine testing for those types of changes yet, but I think over the past few years, there's been a lot of interest in kind of exploring how what we call these epigenetic modifications can influence presentations. And I think that's something that that we think in some cases can explain some genetic differences between identical twins. And so, I think there are other factors that we are really kind of just starting to scratch the surface of that might play into some of the differences that we see.
Great. I feel like I want to go back now and look at my genetics report for my son, because after you explained like how to read some of those pieces, I found that really interesting. And this is another thing that I think is sort of related, and I have the same question. So it says, you talked about interstitial, and the question is, my child's duplication is on the terminal or the end of a chromosome. And is there anything that means in regards to severity? Like, is there anything that we know about that in terms of presentation?
So again, I think the things that we really kind of emphasize are kind of the size and exploring the other gene content. And so terminal duplications are likely to be a little bit on the larger size because they do extend all the way to the end of the chromosome. And so, it would be helpful in some cases, for you to discuss more with your genetic counselor if they could take a look to see, are there other genes included in their duplication that might, be impacting, that might be impacting the phenotype. What we know kind of in general about genes with, MECP2 being an exception, but a lot of genes we know that having an extra copy tends to be a lot more well tolerated than having a missing copy. And so, sometimes it can become difficult because even if we look at the genes that are duplicated, we might be able to say, hey, we know that if this gene were missing, it would cause a problem, but we're not quite sure exactly what it means to have an extra copy of that gene. But, sometimes we can make assumptions, if having a copy missing can lead to different neurologic symptoms. Mm-hmm. It's not a stretch to think that having too much or an extra copy of the gene could lead to different neurologic symptoms as well. And, we're always learning more about new genes, and it's possible that additional genes in that area that are important may be better understood over time as well. So it could definitely be something worth kind of revisiting at intervals, especially if there's a larger duplication present that has, more genes than just MECP2.
Excellent. Yeah, I know. I think I was reading there was some work, you had mentioned this about the IRAC1, that it might be implicated, and there's a group in Spain that's looking into whether that has something to do with our kids being immunocompromised, and if it has other, the two together might really be something, that it might not just be MECP2, it might be also, if you have the IRAC1, then that means additional things, and then we could know more about it. But yep. I think like you said, right, we just don't know yet. Yeah. So, okay. So we have in the Q&A, so the question about the twins, it was that, so according to their genetic testing, they're 90% identical, and they were di-di twins. And so the genetics team had told them that maybe the ovum split early, and that explains why they're not 100% the same. But she wanted to know, oh, go ahead.
Oh, so that, so that 10% difference in their DNA is, could certainly have important, important impacts for sure.
So another question, what's considered a large duplication? So like a 2.9 MB, is that large on the grand scheme of things or small? I know I've been told that like my son, Charlie's is like 300 something. And I think I've been told that that was kind of small, but this one says M instead of K. So right. Yeah, is that larger? That's different, right? You were saying that like
Yeah,
it's usually big.
so kind of 200 or 300 kilobases, would all likely include the MECP2 gene. When you're talking about 2.9 megabases, that means that you're talking about, 3 or 2,900 kilobases. So just an order of magnitude larger. There is no like cutoff per se to say this is a large gene versus this is, or large duplication versus this is a small duplication. And if you kind of scroll back to the slides at some point when you have some free time, there was that one image I showed from the paper that kind of lined up all the different size duplications. And you can see that, even some of the relatively smaller duplications were interspersed with some of the larger duplications in terms of severity. And so, it's kind of a general trend, but again, it's not anything that we can use to make great predictions in any, one person. If we're meeting you and your child when they're a really young baby, we might say this looks on the smaller side. There's not a lot of these other genes included other than MECP2. So we're optimistic that this might be a more mild clinical presentation, but really your child is our best guide, and following them over time is really the best way for us to know how they're going to do.
I feel like I've heard that in many doctor's appointments that, whatever the genetics report says, like we're going to treat the child in front of us. So it really has more to do with like what we're seeing in that clinical presentation than really being able to match it up and say like, well, because it's this size that predicts, this will happen or this will happen, etc. So I think that's helpful to know that it really comes down to what's happening with your own kid, and that can be really different from kid to kid, even with twins. So
exactly.
Okay, so let's see. Here's a question. Can you talk more about X inactivation and why some girls are affected instead of just carriers? And then is there any way to determine whether a girl will be just a carrier or will turn out affected during the reproductive testing that you were talking about? Like, is there a way to know in advance?
So that's a great question. And it's really tricky. So I think one of the trickiest things is that the inactivation patterns can be different in different tissue types. And so we have easy access to testing things like a blood sample or a mouth swab sample, or even in some cases a little bit more invasive, but still relatively easy, a skin sample. The area that we don't have access to testing is the brain, which is kind of the area that we're all really the most interested in. And so in some cases, even though it's a test that could be available, it's not always the most useful. Basically, kind of the way that they do that testing is they look at different markers on a woman's 2X chromosomes to see if one is more preferentially being expressed than the other. There are some cases where the testing is actually uninformative because they just don't have those markers present to be able to distinguish one X chromosome from the other. So there are some people who you order that type of clinical testing and it's just uninformative. And not all labs and not all testing is able to figure out like which X chromosome is which. And so they can tell you one, especially thinking about like the example that was asked about in a prenatal setting, it takes a kind of a high degree of testing to be able to say it's the X chromosome with the duplication that is being skewed or being expressed versus being silenced. And so it kind of becomes a complex degree of testing and not always directly reflective of what's going to be going on in the brain. So I don't think anyone would, there are a lot of challenges to doing that type of testing, and I don't think anyone would feel comfortable, in a prenatal setting using that to draw any conclusions. Unfortunately, I know that would be really useful information for a lot of people to have.
Right, so that if you got the result that you had a girl fetus that did have, something going on with extra MECP2, there's just no way to predict whether that means it's carrier or that means that it would be an affected female.
Exactly.
Yeah.
Exactly.
And that would be, I think, really useful for parents to know. But I guess we're just not, like you said, we've only been doing this for a short amount of time in terms of like the history of genetics. We've only been able to even do these sorts of things for a short
Yeah,
amount of time. So we're just not there yet.
right.
So I
And I
hope we're
think
working on that. Like, I hope somebody out there is working on this though.
Well, I think, I think the benefit is, because, this gene has been so well known for years prior in the Rett syndrome realm, it means that there are a lot of people thinking about MECP2, a lot of people thinking about ways to, intervene and ways that we could potentially target this in terms of therapies. And so, even though the MECP2 duplication is newer to the timeline, there's a lot of groundwork that's already been done, and are many steps ahead from many of the other neurogenetic and neurodevelopmental disorders that are kind of really just recently being described. So, it's an exciting time in terms of research, and there's a lot being done.
Yeah, I did feel people think that this is sort of a funny thing to say, but when we got diagnosed and then you sort of look at it and you see all the scientists who've done so much work on MECP2 gene disorders, and who are, continue to work on it today and trying to, help figure out how could we treat this. It is pretty lucky that there, has been so much already done. We have a long way to go, but there's already a lot of good foundational work there. So here is a interesting question. Can MECP2 duplication syndrome be inherited from the father? And would this always be like a translocation, or does it ever happen some other way?
That is a good question. I'm trying to think of all the scenarios. So, I would imagine there could be some instance, and I would have to really kind of think about this, but I think if it were inherited from the father, it would have to be due to some interesting translocation that was present in him, where the piece of X is probably stuck on another chromosome. I'm thinking out loud here.
Yeah.
I'd say that, one of our mottos in our genetic community is we never say never in the genetics world. So I imagine there has to be some sort of scenario where it could happen if dad has some sort of like very specific rearrangement, but that would be, exceedingly rare.
Okay. Yeah.
And the typical, most of the MECP2 duplications that we see are considered, are likely to be in tandem, meaning they're on that spot on the X chromosome. And those, especially in a male, we know wouldn't be paternally inherited because the boy gets his Y chromosome from his dad, not his X.
Right. Okay. Yeah, that's what I know. I've asked some of these questions before and talking to our doctors and trying to understand what's going on here. And then yeah, that is the answer too. Like, well, generally, but like never say never. So that's really interesting. So let's see. This is a question that I've seen come up a good bit, is that so when you're talking about XQ28 duplication syndrome, is that the same thing as MECP2 duplication syndrome, or how are they different?
It is not. So it is not the same thing. So there is another condition known as XQ28 duplication syndrome that includes kind of a different set of genes. So it's kind of located at a slightly different spot in XQ28 and has slightly different breakpoints. And there are other genes involved in that duplication that can lead to developmental differences and in some cases seizures. Those children don't tend to have as much of kind of the other medical issues that can be present in MECP2 duplication syndrome, but it is its own unique entity. Now, there are likely some children who have big duplications that might overlap both regions, but when you're talking just about the XQ28 duplication syndrome, that typically doesn't include MECP2, and so it's kind of its own entity.
Okay, great. So let's see. If you have time I have just a couple more questions. So Can you talk about like translocation and triplication? Like how common are those? And, are those diagnosed in the same ways that you were talking about with those tests?
So microarrays are pretty dosage sensitive, so they're able to tell if it's looking at a region 1 time, like it's supposed to, 2 times with a duplication, or triplication 3 times. And so a microarray would be able to identify the presence of a triplication as well. It will also identify an unbalanced translocation, but would usually need to have some follow-up testing done. So if you see the piece of MECP2 duplicated, and you also see another corresponding chromosomal region deleted, that's suggestive of an unbalanced translocation, but there would need to be some follow-up testing done to kind of determine the position of everything. So a microarray test will tell you dosage. It doesn't necessarily tell you where the pieces are located, and so that sometimes will require a follow-up test. Usually that test would be called a FISH test, where they kind of use a light up probe that kind of maps to the corresponding spot on the chromosomes and can see where those pieces light up. But usually kind of the microarray test would give you the clue to suggest that there could be an unbalanced translocation if you also see a deletion in addition to the duplication, but then a follow-up test would be needed to kind of figure out exactly where everything is located.
Okay, so it would be additional, but one you could know just from the microarray, and one you would need the translocation, you would need more testing to learn more about where it is and what exactly is going on.
Exactly.
Okay, that's super helpful. All right, so I think you talked about this a little bit earlier, but this is a good question, I think. So if I was a carrier, wouldn't one of my brothers, nephews, uncles, great uncles, etc., have a MECP2 duplication?
Not necessarily. So, in a woman who's a carrier, there's a chance that it just happened brand new in her. In which case you wouldn't expect to see any other family history. It's also possible, let's say it wasn't inherited, and let's say, the maternal grandmother might also be a carrier. Some families just have small families. And so if they just had one daughter, then there just might not be anyone else in the family. Mathematically, it's only a 1 in 4 chance that any pregnancy of a carrier would have the duplication. And so it may just have been flips of coins that, relatives didn't get the duplication. Or, sometimes if it's kind of going way back, babies passed away without workup, without knowing why, could have been attributed to some other finding, could have been affected and just no one really knew. So there are a lot of possibilities, but a lot of times we don't, necessarily see a family history.
Excellent. Well, this has been great. I really appreciate your time. And, we are a little bit over an hour, so I do want to cap it there, but I really appreciate you being here. And I think this was fantastic information. So thanks so much.
And thank you again. It was my pleasure. I really enjoyed talking to y'all today. I hope you found it helpful. And, if there are other questions, Katie, always feel free to email me, or I'm happy to follow up as needed.
Great. That sounds good. Thanks.